Hi everyone. Since we're talking about cells, we're going to take a minute and talk about the cell cycle, which is the growth of cells and the cycle that cells go through when they are about to divide and whatnot. What I have here on the screen for you right here, G1, S, and G2. Collectively, this is referred to as interphase in the cell cycle. Interphase is the part of the cell cycle that cells stay in most of their life. Most of the time, they are in interphase. And in fact, most of the time, they're actually in a phase that's listed, I'll list below shortly, that's called G0. And that's because it comes from G1. If the cell is not getting ready to replicate, then there is actually a stopgap, a checkpoint that sends the cell over into what is referred to as G0, or which is the normal phase, the resting phase for the cell, and it's just going to be doing its normal thing, normal amount of DNA, one whole copy, one whole set of chromosomes from mom, one whole set of chromosomes from the father, and this is a normal amount of DNA for a cell, and that's the way that it spends most of the time. When it comes time for the cell to replicate, it will go from G0 into the S phase. Well, it's called synthesis because of the fact that what's being synthesized is DNA. S is the part of interphase when DNA replicates. Well, why would DNA need to replicate? DNA has to replicate in order for the nucleus to replicate, and then you end up with two identical cells. But so in order to do that, we've got to replicate all the DNA. Otherwise, the two resulting cells would have half as much DNA as we started with. So S phase is when that DNA will replicate. Once DNA replication is finished, the cell goes into what is referred to as G2, or phase 2 of interphase. And at this point, it's still one cell, it's still one nucleus, but it has twice as much DNA as the cell would normally have. Humans, for instance, have 46 chromosomes, 23 from each parent. Well, during G2 in a human cell, you would have twice as many. So instead of 46 chromosomes, you would have 92 chromosomes. Once once all the DNA has been replicated and everything is checked out during G2, the cell's not going to stay that way for very long, because once we're in G2, the cell is committed to division. So it's time to go on into cell division. So here are the parts of cell division. We have mitosis, and then you have cytokinesis. The four phases that I have listed here, prophase, metaphase, anaphase, telophase, these are the parts of mitosis, the stages of mitosis. And then once telophase is over, you actually have two nuclei, and then cytokinesis separates the cell into two different cells. But each cell is identical to the other. We refer to them as daughter cells, and the two daughter cells are identical to the cell that you started with. So prophase, metaphase, anaphase, telophase, those are the four stages of mitosis. Mitosis is the division of, or the duplication of, the nucleus. Mitosis is the division of the nucleus. Okay. Here's that G0. This is where the cell is not going through mitosis, and it's simply doing its cell. And that happens here. We go from G1 into the G0. Once the cell is, once it's time, once the signals have taken place to cause the cell to need to duplicate, we go from G0 into S. That's when the DNA replicates. After DNA is completely replicated, we go into G2 and then immediately into cell division. Cell division, we have prophase, metaphase, anaphase, telophase. That is mitosis. That's division of the nucleus. And then we have cytokinesis, which separates the cell itself into two separate cells. So during S phase, we have chromosome replication. During chromosome replication, and this is a really tough process to kind of explain in a short amount of time, but essentially what happens is you have a complex of DNA polymerases that will attach themselves to the double-stranded DNA. It is not in this chromosome shape that you see right here at that point in time. It attaches itself to the DNA. It simultaneously unzips the DNA, taking the double helix apart, and then builds new DNA on both sides. so that you end up with where there was once just one double strand of DNA, now we have two double strands of DNA that are identical to each other. And so when that happens and it condenses, that's when we get this chromosome shape, because this is already condensed. Before you get this condensation of chromatin, in order to get this chromosome right here, the DNA has long been duplicated. So you get one set on the right, one set on the left, and these are identical. We call them sister chromatids, and they're together to make this one chromosome, and they're connected in the middle with a centromere. So here's a diagram of the whole cycle. So here's the G1, and if everything is just normal and the cell doesn't need to grow and the cell doesn't need to divide, it goes off into G0, where it stays until it needs to divide, and then it jumps back into S. DNA is replicated. Once DNA is fully replicated, we go into G2, which the cell is, everything's normal about the cell except for the fact that now its nucleus has twice the amount of DNA that it should have normally. That's when it goes through mitosis, prophase, metaphase, anaphase, telophase. At the end of telophase, you have two identical nuclei, and then you have cytokinesis, and at the end of cytokinesis you have two cells that are in G1, and those two cells that are now in G1 are identical to each other. In other words, you have two daughter cells that are identical to the one cell that we started with. So if we take a look, G1 cell grows and its volume produces a whole bunch of transfer RNA and messenger RNA. You have ribosomes and other cell components that are replicating. Remember, mitochondria will replicate on their own. There are always going to be mitochondria all over the cell, so when the cell divides, you end up with plenty of mitochondria on both sides. During S phase, DNA replicates. During G2, final preparations are made for mitosis to hit those final checkpoints. If everything's okay, then we zip right on into mitosis, prophase, metaphase, anaphase, telophase. And then cytokinesis puts us to having two cells, each one that are now in G1, and they're identical to the cell that we started with. So here's a description of each one of the phases of mitosis. Prophase. During prophase, all of the chromatin starts to condense, and you end up with what we see as chromosomes. So it starts to condense. If you can actually see chromosomes condensed, you're now in prophase. Once you are in metaphase, you no longer have a nucleus. The nuclear envelope has totally disappeared, and all of the chromosomes are lined up along the center of the cell. We refer to that as the metaphasic plate. Then in anaphase, this gets pulled apart. Telophase, we get new nuclear membranes forming around the two sides. Now, when I say they get pulled apart, the two identical chromosomes get pulled apart, and one whole set, gets pulled to one end of the cell, and the other whole set gets pulled to the other end of the cell. These are centrioles. These structures right here on the ends, they're forming these microtubules, which is called a spindle, that pulls the chromosomes apart. In telophase, the chromosomes are surrounded by new nuclear membrane, and the chromatin starts to unwind and dissipate. And then you have cytokinesis, and during cytokinesis, the new cell membrane forms between the two cells, and you end up with two cells where there was once just one cell. And as in everything else, here's all of the information on the GACE. You need to make sure that you're following through and taking a look at these. Here are all of the steps again. So take a look at those. Make sure you have read all of the description of each one of the steps, each one of the stages. What you're looking at here is actually meiosis, not mitosis. Meiosis is the production of gametes. because we want to end up with cells that have half as much DNA that we started with, because then that gamete, which is haploid, is going to get together with another gamete which is haploid from another individual and create one cell that is diploid and has the full complement of chromosomes, but it's a completely genetically unique mix. So this is meiosis. Okay, it starts out kind of like mitosis, except for the fact that what lines up here in the center during metaphase 1, you have the chromosomes and their duplicates plus their homologs. So what are homologous chromosomes? If we take a look at this second paragraph right here, one of the homologous chromosomes are the chromosomes that you have from each of your parents. See, during mitosis, homologs don't come together at all because all you're interested in doing is duplicating all the DNA. But when we're trying to form gametes, we want to make sure that there's a good mix of DNA from both parents in each one of your gametes. And so the chromosomes that come together are the chromosome, like chromosome number 1 that you got from your mom and chromosome number 1 that came from your dad. They code for the same traits, but they're not genetically the same because one came from one parent and one came from the other. So what you see here lined up, notice that in metaphase 1 you actually have 4 chromosomes that are lined up together, 2 on the left and 2 on the right. These are homologous pairs. Chromosome that came from mom plus its duplicate, and the chromosome that came from dad and its duplicate, and that's true across the board. So the homologs get pulled apart during anaphase 1. You get new nucleus forming in telophase 1. It doesn't stay that way for long. Then we go into prophase 2, where only the duplicates line up, and here we actually have 2 cells because we got 2 cells here. Each one of these, now you have the chromosome and its duplicate that lines up. They get pulled apart, and essentially what you have going on here is a lot like mitosis, where just the chromosome and its duplicate gets pulled apart. We call them sisters. And you end up with 4 cells at the very end. Now notice that we didn't have another duplication of DNA. So you only had 1 duplication of DNA, which means that you have 2 splits even though you only had 1 duplication of DNA. So we end up with 4 cells that have half as much DNA as we started with. Each of those 4 cells is genetically unique, and they have half as much DNA. We refer to them as being haploid gametes. Now, why are they genetically unique? They're genetically unique because up here during metaphase 1, when all 4 chromosomes are together, the homologs plus the duplicates, and so you have 4 chromosomes together. When those 4 chromosomes are together, they actually will cross over each other and it mixes up the genetic material. So you get a good mix of genetic material from both parents in each one of the gametes. But each one of the gametes is genetically unique. So that's kind of an important piece. So that's a big mixing of genetic material, which means that you end up with mixtures of genetic material for sexual reproduction. This process, the process of meiosis, which I want you to go back and I want you to read this thoroughly. The process of meiosis only takes place in the production of gametes. This doesn't take place in any of your somatic cells, any of your cells anywhere else in your body. It only takes place for the production of eggs and the production of sperm. That's it. It doesn't happen anywhere else. So here we can see meiosis is taking place in the testes, and this is where we are producing sperm. Okay, so what ultimately you end up with sperm cells, and then so you end up with these haploid, a sperm cell that is haploid. That haploid sperm cell then is together with an egg cell which is also haploid, and that forms a zygote, which has the full complement of DNA, but it has DNA from both parents, and that goes through mitosis, until you get to growing into a person. So make sure that you go back and take a look at this. So here are just to kind of sum things up, the role of genetics and chromosomes in the transfer of biological traits. Gene, the gene is a portion of DNA that actually determines a particular characteristic. A locus is a particular spot on a chromosome where a gene is located. Typical cells for any organism that was developed through sexual reproduction is called diploid. That means you have two complete sets of your chromosomes, one from each one of the parents. During mitosis, that diploid cell produces two cells that are identical to each other, also diploid. During meiosis, in order to get haploid gametes, you take a diploid cell and it goes through the whole meiotic process that I just explained, and you end up with a haploid gamete. And every cell that goes through meiosis, you end up with 4 genetically unique gametes. So when we think about the way gets inherited, remember, we've talked about the fact that an allele is a version of a gene. A genome is the complete set of genetic material in an organism. Genotype tells me exactly what the genes are in an organism. Phenotype is what an organism looks like as a result of the genotype. Like, for instance, words that we would use to describe a genotype would be homozygous dominant. If you have 2 copies of a dominant gene, you could have homozygous recessive if you have 2 copies of a recessive gene for a trait. We could have heterozygous if you have 1 dominant and 1 recessive for that trait. It only takes 1 dominant gene in order for a trait to show itself. Phenotype is when we describe what something looks like. This would be tall, short, green, yellow. Those words that describe what something looks like would be its phenotype. Gene expression is when a gene is actually expressed as a phenotype. So, and this is what I was saying, you have a dominant allele, which could mask a recessive allele. Typically, when we're writing down dominance, we use a capital letter. Recessive, it takes 2 copies of a recessive gene in order for that to show itself. We usually represent that with a lowercase letter. Homozygous means that both alleles in a particular genotype are the same. Heterozygous means you have 1 copy of the dominant, 1 copy of the recessive. If we take a look at these example genotypes down here at the bottom, 2 capital letters, it takes 2 letters to represent each trait. 2 capital letters would represent homozygous dominant, meaning both copies of the gene are dominant. So this individual got a copy of the dominant gene from each parent. If both are lowercase, that means this is homozygous recessive. So this individual got a copy of the recessive gene from each parent. And then this is heterozygous, meaning you got 1 copy of each. And if this is a simple dominance, then whatever the dominant trait is what would show up here, because you've got 1 copy of the dominant gene. So if free earlobes is the dominant characteristic, then homozygous dominant shows free earlobes. Heterozygous shows free earlobes, and it would take homozygous recessive in order to show the recessive characteristic, which would be attached earlobes. Gregor Mendel was an Augustinian monk who bred pea plants, and he came up with all of these different mathematical models that we're using in order to work through genetic traits. he did study one trait at a time, and as it would have it, all of the traits that he studied were not allelic, meaning they weren't on the same they weren't on the same chromosome, and they were not gene linked in any way. And so he came up with some really what are pretty simple ratios that will describe the way that inheritance works. We know that they don't always work, because there are such things as gene linkage, where two genes are actually linked together and they don't separate by pure chance during meiosis. So, laws of heredity, we call them laws, but really they're more like principles, because there really are no laws when it comes to biology. The dominance, if you have simple dominance, all it takes is one copy of the dominant gene for it to show itself. Segregation, the law of segregation, or the principle of segregation for Mendel, is that if, for instance, if this is the parent cell, one copy of each allele will go to each gamete, and that is determined completely by chance. So 50% of the allele of the gametes will get the dominant allele, and 50% will get the recessive allele, in this case, if you had a heterozygous parent. The law of independent assortment, you have to have more than one characteristic in order for there to be the law of independent assortment. But if we were looking at two characteristics, like the, say, a green versus yellow pea, and then tall versus short pea, and we bred these together, if the tall short separates independently from the yellow green, then we should have tall green pea plants, tall yellow pea plants, short green pea plants, and short yellow pea plants, as long as they are not linked in any way. But that would be independent assortment. Those two traits sort independently of each other. So if we were to work a single factor cross, we use what is referred to as a Punnett square. And so if we were looking at, say, for instance, something with Tourette syndrome, if you had homozygous dominant for Tourette syndrome, the heterozygous would have Tourette syndrome, and lowercase is homozygous recessive would be normal. Then what we would have to do is we put together our Punnett square. And if we have two parents that both have Tourette's, but they're both heterozygous for it, one parent having one copy of each allele, and then the other having one copy of each allele. Then we fill in the square. This possibility would be two copies of the dominant gene. This would be a copy of the dominant and a copy of the recessive. This would be a copy of the dominant and a copy of the recessive. And this would be a copy each of the two recessive genes, which would mean that there's a 25% chance that each offspring would be homozygous dominant. There is a 50% chance, or 2 out of 4, that each offspring would be heterozygous, and then there is a 25% chance that for each offspring that they would be homozygous recessive. When we think about more complex models, phenotypes are always determined by alleles. They can interact with each other, and they can also interact with the environment. Besides simple dominance, we could have X-linked characteristics or sex-linked characteristics, meaning that they're traits that ride around on the X chromosome. Well, if we think, if it rides around on the X chromosome, if it's a recessive trait, then it's more likely to show up in males, because males only have one copy of the X chromosome. Males have a copy of the X and a copy of the Y. Those two things are not homologous to each other. So if there's a problem with the X chromosome in males, it's going to show up. Whereas with females have two copies of the X chromosome. So if it's a recessive trait, they're going to have to get a copy of the recessive trait from both parents in order for it to show up. So typically sex-linked traits show up more often than not in males. just because it's more difficult for you to get both of the recessive traits in females. Codominance means two traits work together and each one shows itself. Incomplete dominance is when traits actually mix. Think about the difference between these two as incomplete dominance is like mixing paint. If I have red paint and white paint and I mix them together, I get pink paint. And that would be incomplete dominance. Codominance would mean if I had red paper and I ripped it apart, and I had blue paper and I ripped it apart, or white paper and ripped it apart rather, and I make a collage, I can still see very clearly the red paper, I can still see very clearly the white paper, even though it makes a new picture. But each one of those genes is expressing itself, and that's called codominance. Multiple alleles. Most characteristics are polygenic, meaning that it takes more than one gene to control it, like eye color, hair color, that sort of thing. So, and I've already explained this about X-linked genes or sex-linked traits. And also, I just described codominance. So if you think about codominance, if things are codominant, you're going to see both of the genes being expressed. Incomplete dominance is like mixing paint. So if you have a white rose and you cross it with a red rose and you get pink roses. That's incomplete dominance. So, and that's what you have right here: a white flower, red flower. Mix them, and when you crossbreed them, you get a pink flower. That is incomplete dominance. Multiple alleles. Most genes are controlled. Most genes have control or are controlled by multiple alleles. So you've got multiple alleles that could be a possibility. But any one organism can only have two particular alleles. An example of both multiple alleles and codominance would be blood type, because there's actually only two alleles. There's A allele and there's B allele. O is not a separate allele, but it's the absence of the other two proteins. A and B will show codominance, and both is dominant over O. The only way that somebody has O-type blood, meaning that they don't have either of these two proteins, is if you've got the lack of the A or B protein from both parents. If anybody with blood type, if you have blood type A, that means you could be A or AO. If you're B, you could be B or BO. You could also be, if you're A, you could be AA. That just means you still have the A protein. If somebody's BB, that means they only have the B protein. If somebody has an A from mom and a B from dad, that means you're type AB. That means you produce both blood type, both proteins in your blood, the A protein and the B protein. So that's an example of multiple alleles. And so that's what I'm showing here. The I, that's neither A or B. So we could have blood type A if you've got two A's, blood type B, A if you have the A and then the lack, and then B if you have two B's, or B and then the lack of it, and then if you have A and B, you have blood type AB, and if you have the allele that doesn't code for either type of protein from both parents, then you have type O blood. Now, how does a gene work? Okay, DNA codes for RNA, and RNA is translated into a protein. And this is a huge, trying to put this together on one page doesn't do it justice. Protein synthesis is a huge thing to try to explain. You can read this. We take a look at what's going on. DNA. I don't like the fact that this diagram has DNA replication right here, because DNA replication is not part of protein synthesis. DNA replication is part of cell division. DNA replication doesn't have anything to do with protein synthesis. From here, this second DNA right here, moving to the right, that's protein synthesis. DNA is transcribed into RNA. RNA is translated at the ribosome into a protein, or the RNA itself is the instruction that the ribosome uses to put together a protein. And then that protein itself, it could be whatever the protein is. There are any whatever a protein is. It could be an enzyme that is going to control which parts of the DNA are actually expressed later and whatnot like that. So a gene, which is a segment of DNA that codes for a particular protein. That gene is transcribed into RNA in the nucleus. That RNA leaves the nucleus and goes to the ribosome. The ribosome then reads that RNA transcript and puts together a protein from amino acids. So the RNA tells the ribosome essentially which amino acids to put together and in what order. And it puts together a sequence of amino acids, and that sequence of amino acids is what makes a protein. DNA itself is made of adenine, guanine, cytosine, and thymine. These are nucleotide bases. The pattern of the sequence of adenine, guanine, cytosine, and thymine that you have in the DNA is what codes for particular amino acids. RNA, which is ribonucleic acid, which is the transcript, is exactly the same with adenine, guanine, and cytosine, but there is no thymine in RNA. What you have is uracil. Bases are complementary. Adenine always pairs together with thymine or uracil. Cytosine and guanine always pair together across the DNA strands. So A and T always are paired together. G and C are always paired together. Those are complementary base pairs. So a monomer, which is a nucleotide for DNA, is composed of the sugar, deoxyribose, a phosphate group, and then one of the four bases. The deoxyribose, the sugar, and phosphate form the backbone. Think of a ladder. They form the sides of the ladder. And then the bases, adenine, guanine, cytosine, and thymine, are the steps in the ladder, and they are coming together across the strand. DNA is double-stranded, RNA is single-stranded. Here are the bases. These are nucleotides. So this would be a nucleotide with thymine. So deoxyribose. So here's the sugar and here's the phosphate. And it's actually the phosphate. The phosphate of one connects to the sugar below it. So the phosphate here will connect to the deoxyribose of the sugar, actually to the sugar above. And then this will connect to the phosphate that's directly below it, and that's what forms that sugar phosphate backbone. And then you have the nitrogen base here, and so it is that sequence of the bases, adenine, guanine, thymine, and cytosine, that essentially determines what amino acid is put into place, which means that determines what protein is being made. And it takes 3 nucleotide bases to code for one amino acid. So DNA is a double helix. It resembles a twisted ladder. It has a sugar phosphate backbone that makes the two sides. The two sides are complementary, with A always pairing with T and C always pairing with G up and down the ladder. Hydrogen bonds hold the two sides of the ladder together. As you can see right here, 3 hydrogen bonds hold together cytosine and guanine. 2 hydrogen bonds hold together adenine and thymine. So essentially the genetic code works like this. DNA is transcribed into RNA. RNA is then translated into a protein. And this is the genetic code. This is the way the genetic code works. That would be referred to as central dogma. And so that's kind of that's the way that works. DNA codes for RNA. RNA is coding for a particular protein. So the DNA ultimately controls what protein is being made. Now, DNA replication. DNA replication does not have anything to do with protein synthesis. I'll say that one more time. DNA replication does not have anything to do with protein synthesis. DNA replication has to do with getting a cell ready for mitosis. And this I actually described to some degree when I was talking through mitosis.